Lici Schurig Briccio, and I'm a research scientist in the Biochemistry department, and I teach Biochemistry 455, which is Techniques in Biochemistry and Biotechnology. And this course is currently only taught during the spring.
Can you give me a brief overview of what is covered in your course? What are the learning goals?
So this is a laboratory course where students will learn different basic biochemistry laboratory techniques. They go from simple techniques like PCR cloning to more elaborated like ELISA and EMSA. And we emphasize the learning of different experimental methods, data analysis, and good laboratory practices, including safety. Also, we emphasize the teamwork. So in this course, students, they need to work in pairs through the whole course and help each other. Some important goal also of this course is to learn how to troubleshoot their experiments. So students, they need to figure out what probably went wrong and fix it or repeat it. And in order to do that, they need to learn a lot of the theory behind each of the techniques that they perform during the course.
What is the format of your course?
So this is a 4-credit course. So students, they have 8 hours per week in the lab, and then 1 hour per week of lecture.
How are your students graded in their course?
Well, we have pre-lab homeworks that they need to be submitted before the lab actually starts. Then they have lab reports and 2 exams. So they have a midterm exam, which is a lab practical, and they have a final exam that includes a presentation of a project that they have to come up with, and it should include some of the techniques that they learned during the course. And this final exam also includes a written test.
Okay. Historically, which portion of the grade breakdown do students struggle the most with?
Well, the written test. The final exam written test. So I think why is that is because a lot of the questions we ask in there that involve problem solving and data analysis are very hard for students. So we are planning to include some of those questions in the lecture so students get time to practice on those.
Are there points associated with attendance?
No, they are not, but this is a lab course. So students actually need to be in the class to learn things, right? They need to practice. So labs are required. They can miss 2 of them, but after that they need to drop the course. And lecture also, there's no points associated, and it's not required, but it's highly recommended, especially because we explain in detail, I explain in detail what is going to be, like what the students are going to be doing during the experiment, what are the key things that they need to know. And sometimes also we need to start soon the lab, so lectures are just before the lab starts. If we need to start soon with the lab experiments, sometimes my lecture is shorter, so we recommend students to come to the lecture for sure.
Are there any required textbooks for this course?
No, there is not. The reason is because we added all the relevant information to the protocols, so students, they have the information they need there. But we suggest some textbooks that if they want, they can go and go in more detail.
What kind of background knowledge do I need to take this course? Are there any prerequisites?
Yes, they are. So some of the courses that are required are some basic chemistry courses like Chemistry 232 and 236. Also MCB 251 or some equivalent course, and MCB 354 or MCB 450 or equivalents, yes.
How do topics covered in your course apply to current research, whether it's medical, science, or scientific?
most of the techniques taught in this course, they are used actually in medical diagnostics, in quality control in industry, and in basic research. So, yes.
Okay. What opportunities do you provide outside of class for students to ask questions or get assistance?
So there's office hours. Currently there's 4 TAs that they will be offering 1 hour per week office hours, and myself 2 hours per week. If the students they cannot make it, they can contact me by email and we can set up another day to meet.
What is your favorite experiment that the students perform?
Okay, so I don't think I have a favorite experiment, but the one I think that includes a lot of relevant information and a lot of important concepts is the protein purification lab. And that's because in this one they actually apply and I think understand better what gene regulation and gene expression means and how useful it is. Also, they can see different ways of protein purification and how they can take advantage of protein-protein interaction and the use of reporter proteins. They can also be able to see if a protein that they isolated is pure enough or not, and they can identify it and quantify it as well. So it has a lot of important concepts.
How helpful do you feel this lab material is in helping students understand other courses in the MCB curriculum?
I think it's very helpful because a lot of the things that they've learned before, like I said in MCB 251 or 354 and 450, they will be applying all of that. You know, it's like they've learned about enzymology. Here they will do enzyme characterization, and they will measure kinetic parameters by themselves. So it actually correlates a lot.
Are there lab practicals in this course? If so, what is the format?
Yes, we have a midterm lab practical, and it has 2 questions. So the first question, it's for to evaluate a student's ability or skills in the lab. So they will perform a short experiment and provide the results. And the second question is about analyzing data. So students we will provide the data and they will have to analyze it.
How would you describe the workload for this course? Is there an estimate for how much time students will spend outside of class?
Well, that will depend from student to student. For some, it might take more time than others. It's a very intense course, for sure. And I think one of the things that takes more time for students to develop is the lab reports. But they can still work on them during the lab class, especially because some experiments, they have these incubation times where they need to wait, right? So they can work on them during that time. So it's possible.
Is there anything else that you would like to tell us about your course?
Okay, so I started teaching this course last spring, and I got a lot of feedback from coordinators, TAs, students. So we've been trying to simplify this course in a way that will help students to understand it better. And in doing that, what I did basically is remove all the repetitive experiments and organize it in a way also that the students will see a connection between each of these labs. I encourage the students to look at the syllabus and see how this course looks at this moment. We are going to continue making changes, and yes, I hope they decide to take it.
Thank you very much.
Yeah.